Hi everyone, my name is Duncan McGee. I'm currently working at Dr. Tseytlin's lab at the Health Sciences Center, and I've been optimizing a 3D bioprinter for fresh bioprinting. So our goal was to implement and optimize a 3D bioprinter that would be able to print designs using a high resolution. So basically, bioprinting can be used in tissue engineering to replicate human body parts like bones, blood vessels, and other natural tissues. So bioprinting is just 3D printing, but instead of plastic, it uses a bioink. In this case, it's alginate, but it could be collagen or some other synthetic or organic material that's able to serve as like a scaffold for living cells during the printing process. So in this case, the 3D bioprinter will utilize extrusion-based bioprinting, as you can see in figure one. So in our case, a 3D plastic printer was modified to print with alginate using fresh, which is the freeform reversible embedding of suspended hydrogels. And basically what this means is there's a gelatin support bath that has small gelatin beads in a calcium chloride solution. This calcium chloride solution allows for the alginate to polymerize through ionic cross-linking during and after the printing process. So our goal was to increase the resolution of the print and be able to print. And to do this, we had to optimize the print settings. And in the end, we were able to print an alginate tube that modeled an artery. And to do this, we had to optimize the extrusion rate, print speed, and other settings using Ultimaker Cura, which is a 3D printing software. One of the biggest increases in the resolution of the print was switching from fresh version 1.0 to fresh version 2.0, which decreased the size of the gelatin beads and made it more uniform. So as you can see in figure 3 on the left, through a microscope, there's some large chunks of gelatin and it's all ununiform, while on the right, there's a bunch of small particles of gelatin. So this allowed for there to be a high resolution in the final print. So using the fresh 3D bioprinting, we were able to successfully print a tube and flow a red dye through it, just to prove that it was viable. And our ability to successfully bioprint an alginate tube shows that bioprinting could possibly be used in the future to replace animal testing, as you can replicate human tissue. So in the future, what we're going to do is print either an alginate tube or another design and image it using electron paramagnetic resonance. This is better known as EPR. So EPR allows for the 3D mapping of oxygen and other essential for life molecules. And so we can then image the changes in oxygen distribution across the tube. And this allows for the oxygen diffusion rate from the inside of the tube to the outside of the tube to be measured. And this will be like a proof of concept experiment to prove that bioprinting is a viable option in the future to replace animal testing and that it can replicate human body parts like blood vessels, bones, and other tissues. Thank you.